Hi class, we're moving on to our next unit, genetics. Genetics is a fascinating unit of study that talks about why different organisms look the way they do, maybe sometimes act the way they do, how the different organ systems function together, things like that. So when we're talking about genetics, it's how you inherit your specific characteristics. So for my instance, why I have two girls, one has blue eyes, one has green eyes, and how they actually inherited those. Okay. When we're talking about genetics, we really need to understand the backbone of genetics, which is going to be your DNA. DNA stands for deoxyribonucleic acid, and you might have talked about it a little bit in grade 10 when you were looking at your mitosis. Okay, so what does DNA do? It controls everything that goes on in the cell. It controls how your organs and your organ systems function together. It controls what you actually look like. So it's kind of the blueprint of our cells in our body. It stores that genetic information, it codes for a protein, and then those proteins allow for our body to function. We don't really look this year on how DNA goes to a protein. That's more in the grade 12 course. But that DNA is actually found in a nucleus. So in that membrane-bound nucleus, that's where you're going to find your DNA. When cells undergo cellular replication, they have to duplicate the genetic information so that each cell has the proper amount of DNA, and that's through that process of mitosis that you would have learned about last year. We'll do a quick review about it in the next lesson as well. And then if we were in class, I'd talk about how look to your left, look to your right. You are 99.9% similar in your DNA to the people around you. You are only 0.1% unique from the person beside you genetically. And why you are so similar to somebody that maybe looks completely different than you is that your bodies do the exact same thing. Your cells do the exact same thing. So that 99.9% similarity is based on telling your cells how to function, telling your organs how to function, and telling the organ systems on how to work together. So that 0.1% is what makes you unique and what makes you special. Okay, so how did we figure out that this DNA was in our cells? There's many scientists that were involved in this process. We're only going to highlight 2 or 3 scientists. Actually, I think there's maybe 4 scientists that kind of go through it. Grade 12 will go over even a couple more. Okay, so when we were looking at DNA, they had to figure out how is this genetic information getting passed from generation to generation. And so scientists came before Rosalind Franklin and Watson and Crick were able to kind of figure out that it was in the nucleus, that it was this DNA molecule, but they didn't actually know what the structure actually looked like. And that's where these guys came into play. So Rosalind Franklin, she used this X-ray crystallography. So it was a way to take a picture of the molecules of DNA. And so you can see it here in this photo that she was able to take a picture of your DNA, and it kind of looks like a little crisscross. But what that's showing is that the DNA was coiled. It was that helical structure, and she was able to come up with the rough size of it. From there, as well as using other scientists' kind of research, Watson and Crick came in. And so they were able to take her picture and everybody else's data and actually make a model. And you can kind of see it in this picture. So that's going to be the model of DNA that was like widely accepted. And they won their Nobel Prize for it. And so they were able to take they didn't really do the research as much themselves and figuring everything out, but they were able to take everybody's data and put it together. And so between kind of these 3 scientists, they were kind of getting the final piece of what that DNA molecule looked like. And what was determined for the DNA molecule structurally is it is made up of nucleotides. They're going to be this building block that makes our DNA. The nucleotide consists of a pentose sugar. Pentose just means 5s. So it means 5 carbon sugar. And so this is going to be your 5 carbon sugar. Okay, and so that 5 carbon sugar in DNA is known as deoxyribose. That's your pentose sugar. In DNA, RNA, which we'll get to a little bit later, is going to have a different name. Then you're going to have your phosphate group. And if you remember from chemistry in grade 10, phosphate is just PO4. Okay, and then your nitrogenous base. And what that means is it's going to have lots of nitrogen in its structure. So this is kind of the simplified one at the top. And then at the bottom, you can actually start to see your molecules. And so let's look at your deoxyribose sugar. So there's a carbon here, 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 and here. So you have 1, 2, 3, 4, 5 carbons, which makes sense with the pentose carbon, 5 carbons. Phosphate is our PO4 molecule here, and then the nitrogenous base is just your ring structure here with lots of nitrogen. Right now, we can see it as 2 rings. It can be 2 or 1, and we'll go through that next. And then they're held together by covalent bonds. And if you remember, again, from 9 and 10 chemistry, the covalent bonds are 2 nonmetals. Okay, so now we have those nucleotides. We are going to put those nucleotides together. And if you kind of see in this picture here, ooh, I missed the phosphate. Let me grab that in. There's a nucleotide, there's a nucleotide, and you have nucleotides. And so those nucleotides are going to make up your DNA. And if you actually notice in the picture, I'm just going to erase everything, you have a strand here, and then you have a strand on this side as well. Those are going to be your 2 strands. And if you actually look, this side it says 5, this side says 3, this one says 5, this one says 3. So they run in opposite directions. That's really important to remember, especially for grade 12. This is what's called they run in anti-parallel. So one runs in one direction, the other runs in the opposite direction. Okay? And so you can kind of see that the OH or the phosphates are in the opposite direction, and how they kind of base it on is based on your hydroxide groups on that pentose sugar, and that's how they kind of come up with the numbering. You can see they kind of have the numbers there. We're not going to worry about that this year. Next year, we'll talk about why those numberings are going to be important. Okay, so now we have our phosphates and sugars and our nitrogenous base pairs. Remember I said the nitrogenous base pairs can change, and that's what we're going to go over next, is how they actually are different. So there's going to be 5 nitrogenous base pairs, 4 for DNA, 4 for RNA. The 3 of them are going to be in both. One's going to be in each of the opposite. So you're going to have adenine and guanine. They are going to be known as your purines. They are a double ring structure. And if you look here, you can see the double ring means that it has a ring attached to a ring. Okay, these are going to be found in both RNA and DNA. And we'll go through that a little bit later. Okay, then you have your pyrimidines, which are going to be your single ring structure. So you can see here, you just have the one. And they're going to be comprised of cytosine, thymine, and uracil. This guy here is only found in RNA, and RNA is just your single-stranded version of your DNA. We kind of mentioned it back in the diversity unit as well. Okay, how do we know which ones of these nitrogenous base pairs actually like to bond together? This is what's known as Kharkev's rule. And so what they were able to determine is adenine always pairs with thymine, and so I always remember at goes together. If you can remember at, so AT goes together, then you always know C and G go together as well. So cytosine always pairs with guanine. And if you look at the picture here, you can see adenine and thymine always go together, and what they're actually forming is that they form 2 hydrogen bonds. I'm just going to write H bonds just so I'm going to run out of space. So those are going to be your 2 hydrogen bonds. Where if you look at cytosine and guanine, they are going to form 3 hydrogen bonds. We have a little more space so I can write the words out. And so that's what it causes them to only be able to bond together, is the way they can actually fit their hydrogen bonds together. So now we have this nice, beautiful strand of DNA. You have your deoxyribose sugars, your phosphates, your specifically bonded nitrogenous bases, but it's like 1.8 meters long in every single cell in our body. That's not practical to just kind of have this long strand. So they have to have a way to organize the DNA and make sure that it fits within the cell's nucleus that it needs to. And so what it has to do is it kind of has to keep coiling and coiling and coiling. So if we look at the picture here, you can see there's your double-stranded DNA. It's going to coil around what's called a histone. And then those little guys are going to form your chromatin fibers and then a supercoil, which will then become your chromosomes. And so from there, our cells will have all these little nicely packed chromosomes of DNA. If we're looking at all of our body cells, we're going to have 46 of these chromosomes. If we start looking at the sperm and egg cells, you're going to have less, and we'll go through that in the next lesson. Okay, so when we're talking about chromosomes, it's just your coiled DNA containing all of our specific genes. And as I said in the last slide, we have 46 chromosomes, but they are arranged in pairs. So here it's just showing that you have one copy of each. So this is more like your egg and sperm cells, but normally in a normal like skin cell, your muscle cells, you would have another pair. Bear with my drawings. So you would have your two pairs. And so that would be your pair of chromosomes. You would have one that came from mom and one came from dad. So of these pairs, one is from mom, one's from dad. And the same thing as you would have them paired up. Every species will have their different amount of chromosomes. Dogs have 78, cats have 38, chimpanzees have 48. And so that's why also evolutionary, we are compared very similar to chimpanzees because they have 48 chromosomes and we have 46. And we'll talk more about that in the evolution unit. So when we're talking about chromosomes, remember how we said that they have in pairs? When we're talking about them being in pairs, that's what's known as diploid. It means there's two sets. That's a super important word to kind of know because we're going to need to touch on that throughout the next couple lessons. So it means 2N chromosomes. So for every like chromosome 1, you have 2 copies. Chromosome 2, you're going to have 2 copies. Okay? And as I said in the last slide, one from mom, one from dad. The sperm and egg cells are going to be different. They are only going to have one set, and that's what the picture in the last slide showed. And that's what's known as haploid. The reason we do this is if you think about your sperm and egg cell having 23, 23. Once fertilization happens, it brings you back up to 46. So it gives you a healthy set of numbers. If they had been 46, 46, that means the offspring would have 92. That doesn't work. So that's the reason why your sperm and your egg cells need to be haploid. Okay? And then sometimes it can happen in humans. It will happen in other species as well. You can have 3 or more copies of a gene. In humans, it ends at kind of 3. Anything more, usually the offspring won't survive. In plants, it can be so usually it's 3 or more. They're called polyploid. Where we see this in human is usually in trisomy 21 or Down syndrome, and means that we have a 3rd chromosome. Sometimes you will have one less copy of chromosomes, and we'll talk about that in a later note as well. Okay, so we talked about the idea that your chromosomes contained our genes. Our genes aren't the genes that we wear. They're the genes that code for specific traits. And so this is why you have your color eyes. This is why you might look more like one parent, or this is why your siblings look identical to you or not so much. And so that has to do with those genes that code for specific traits. They are located on specific chromosomes and specific parts of the chromosomes. Okay, and so we'll go through about different traits later. We're not going to look at which eye color trait is on which chromosome. That's just getting kind of a little bit higher up levels of stuff. So we're not going to go that far, but we'll talk about different traits that we inherit through our genes in the coming notes. Okay, perfect. Thanks, class. We will keep tracking with the worksheet, and then we're going to move on to mitosis and meiosis.